Hello everyone, hope you all are doing well. So today we are going to do topic 6 of paper 2, that is linkage and crossing over. So first we will see what is linkage. What is linkage? So the linkage, it is the tendency of genes to remain together, tendency of genes to remain together, in their original combination, during inheritance. So what is linkage? It is the tendency of genes to remain together in their original combination during inheritance. While in the inheritance, the genes remain together in their original combinations. Now what are the types of linkage? Types. So first is based on crossing over. Types based on crossing over. So based on crossing over, we have got two types. One is complete linkage and one is incomplete linkage. One is complete linkage, one is incomplete linkage. So in complete linkage, there is complete absence of crossing over. There is absence of crossing over, complete absence of recombinant types due to absence of crossing over. Here crossing over doesn't occur, absence of crossing over. So it can be seen in males of Drosophila, females of silkworm can be seen in males of Drosophila and females of silkworm. Whereas in incomplete crossing and incomplete linkage or partial linkage, it is also incomplete or it is also called partial linkage, there is some frequency of crossing over occurs. It is complete absence of crossing over. Here some frequency of crossing over occurs. It can be seen in mage, pea, female Drosophila, etc. Now the second type is based on genes involved. Now second type of linkage, that is based on genes involved. Here we have got also two types. First is coupling phase. Genes, what kind of gene is involved? That is dominant or recessive genes, involvement of dominant and recessive genes. The first here we have got coupling phase. Coupling phase, here dominant allele or some chromosome and recessive, there are dominant allele here, dominant allele is on some chromosome and recessive on some chromosome and recessive on some chromosome. So dominant alleles are present in some chromosome and recessive in some chromosomes. You can see here, just see in this here dominants are present and here recessive are present. So this is coupling phase. Now second one is repulsion phase. Here dominant alleles in some genes are linked with recessive on some chromosomes, So here we can see like this TR. Here the dominant alleles are linked with recessive, or you can say TR. So this was based on genes involved. Now moving on, The third type is based on chromosomes involved. Third type is based on chromosomes involved. Here there is involvement of chromosomes. We have got two types. That is here we mainly see the location of genes, and it is of two types: autosomal linkage and allosomal or sex linkage. So it is located in autosomes, that is other than sex chromosomes. It is located on autosomes, autosomal, as the name suggests, autosomes, that is other than sex chromosome, and it is located on allosomes, that is on sex chromosomes, that is either X or Y chromosome. So this was the types of linkage. I hope you understand it. Now moving on, detection of linkage. How will you detect the linkage? Detection of linkage. So the most common method to detect the linkage is test cross. Through test cross we can detect the linkage. In this method, F heterozygous at two loci is crossed to double recessive parent. In this method, F heterozygous to two loci, that is AB by AB, each crossed to double recessive parent, each crossed to double recessive parent, that is AB by AB. This is double recessive, one recessive, double recessive parent, and the phenotypic ratio of test progeny. And after the crossing we study the phenotypic ratio. Phenotypic ratio. So what happens if the phenotypic ratio of test cross progeny shows 1:1:1:1:1? It indicates absence of linkage, means the linkage is not present here. If the phenotypic ratio is 1:1:1:1 of parental and recombinant genotypes, ratio should be of parental and recombinant genotypes, then there is no linkage. And if the ratio deviates from 1:1:1, if it deviates and there is deviation in the ratio of 1:1:1:1, then we can say that there is presence of linkage. Linkage is present here. So this is how we will be detect linkage, and detect linkage through test cross. If the ratio is 1:1:1, then there is no linkage, but if there is deviation from this ratio, there is linkage. I hope you understand this. Now moving on, what is the significance of linkage? Significance of linkage. It doesn't permit breeders to bring desirable characters. It doesn't permit breeders to bring desirable characters. Desirable characters in one variety. Here the breeders find it very difficult, both plant and animal breeders find it difficult to combine various characters. Very difficult to combine various characters. It reduces the chances of recombination of genes, reduces chances of recombination of genes, and thus help to hold parental characters, and thus helps to hold parental characters. Hence it helps an organism to maintain their parental, racial and other parental characters, as genes are what do you say, genes are located closely with together. Genes are in good remain together in original combination. They are related very closely. Now moving on, as the linked genes are not inherited together every time, linked genes are not inherited together every time chromosomes exchange homologous genes. Okay. Linked genes are not inherited together every time chromosomes exchange homologous genes during meiosis. So I hope you understand. It helps to hold parental character. It's very difficult to for plant breeder to change the parental character into desirable characters, and here the linked genes are not inherited together every time, and the chromosomes exchange homologous genes during meiosis. Now moving on, what is crossing over? This was the significance of linkage. I will move into the next topic of this session, that is crossing over. It is a random exchange of DNA between two non-sister chromatids, of homologous chromosomes. So it is a random exchange of DNA between two non-sister chromatids. In linkage, we say how DNAs are closely linked with each other, but in crossing over, they are not closely linked. They exchange their DNA between two non-sister chromatids of homologous chromosome. Now, what is the significance of crossing over? It produces new combination of traits or new traits. Through crossing over segments of homologous chromosomes, are interchanged and hence provide origin of new characters. Crossing over plays an important role in field of breeding, very important role in breeding, to improve varieties of plants and animals. So this was all about crossing over. Hope you all understand this. Now moving on. Hope everything is visible. So it was asked in some terms were asked in previous year questions, so we will discuss here. First of all, what is sex linkage? So we just discussed sex linkage. We will just discuss sex linkage. It describes sex specific patterns. It describes sex specific patterns of inheritance and presentation when a gene mutation, inheritance and presentation when gene mutation or allele mutation is present on a sex chromosome rather than non-sex chromosome, is present on sex chromosome rather than a non-sex chromosome. It's not present on non-sex chromosome, it's present on sex chromosome. Example, you can see such as hemophilia. red green color blindness, red green color blindness, some high blood pressure genes. Some high blood pressure genes is often seen in some months. So this was about sex linkage. Now move on to next one. What is autosome? It is a chromosome that is not sex chromosome. It is non-sex chromosome. That is, it is not sex chromosome, non-sex chromosome. The members of autosome pair is diploid cells. These the members are diploid cells and have same morphology with same morphology. While those in allosome pairs which may have different structures. While in autosomes they have same morphology, but in allosomes, that is in sex chromosomes, we can see different structures. And the DNA of autosome is known as autosomal DNA, ATDNA, or AUDNA, autosomal DNA. And humans have 22 pair of autosomes and 1 pair of allosomes. That is 1 pair of sex chromosome and 22 pair of non-sex chromosome, that are autosomes. Now moving to the next term, that is double crossover. What is double crossover? So double crossing over is when recombination during meiosis happen twice in the same place. Recombination during meiosis happen twice in the same place. It is very much rarer. It is very much rarer than single crossover. Single crossover is more common than double crossover. Double crossover is very much rarer. And the double crossover offsprings, the offsprings produced from double crossover, should have same phenotype as parent. Have same phenotype as parent. So we will see how double crossover happens. Suppose these are two non-sister homologous chromatids. Let it be WMBR. So what happens? This is colored, this is non-colored, and they are crossing over here. This is non-colored one and it is colored one. Sorry, this is colored one and this is non-colored one. So what result we will get? Like this. What result we will get? So this portion will break and this portion will break. So here see, this one will become colored and this one will be non-colored, and here we will see this portion. This is this portion, these two are colored and here non-colored, and this is W, this is BR, this is M. So this is what double crossover is. You can see how crossing over takes place. So I hope you understand linkage and crossing over. So that's all for today's session. For any queries, do comment, keep making notes, keep value adding to your notes. If you like it, please press the like button, do share and subscribe. That's all for today. Have a nice day. Thank you.